Okay? Is there anybody who has been unable to access the videos by at least either the icon or the Google video? If you've tried both of them and failed. Anybody? Okay, good. Just a little bit of practical stuff, and we're probably going to be talking about this more than once, just to make sure that everybody's all on the same boat, on the same page, rather. We have a fairly large class. Obviously, not everybody is here today. But we'd have to sit down together to take exams. And so we have 2 exam rooms for this semester, and we have to divide people up between those exam rooms. It creates chaos to just simply say, oh, go somewhere. So you are assigned to an exam room based on your last name, and I have a table here of where those different places are. The larger number of people will simply be here. If you're somewhere in the middle or end of the alphabet, you're going to be somewhere else. So if your last name comes through A through M, you'll be here for this exam. And N through Z, you'll be at the other auditorium. It's right sort of catty corner across the passageway. We ask you to bring some kind of ID with you at the very least, because we're only going to seat the people who can show they're at the right place right away. If you don't have any ID, we're going to ask you to wait until we've seated the students who have ID, because we don't have an unlimited number of seats for people, or an unlimited number of exams and seats for people. On the way out, when you're turning in your answer sheets, we're also going to ask you to show something that has your university ID number on it, either your university ID card or a printout from your ISIS account, something like that, because we're going to check that you've entered your ID correctly. This sounds like a really simple concept, but what we have found is that if we don't check, we get anywhere from 25 to 30 errors in that number, and each error takes 3 to 5 minutes to fix. If we check the IDs, they're almost all right, and we can correct them right away. So have something with your university ID number on it. We'll have to hold your answer sheet separately if you don't have that. So the simplest thing is to bring your university ID with you for the exam. Please make every effort to arrive promptly. If you're not here before 3:30, we will ask you to wait outside, and we will admit everybody who's late at the same time at 3:40 with additional time, so you're not caught short for time. Just try to minimize the disruption to people and so on. There are a couple of students, not many, who have some kind of a conflict with their laboratories who may need to be seated between 3:40 and 4 o'clock. Please be sure that I have your name and can make arrangements with the proctors so they know about it. Otherwise, you won't be admitted. I'm sorry to be so formalistic about all this, but when we have large numbers of people in 2 places, we have to try to be as organized about it as we can be. Do people have questions about just the mechanics of the exam so far? You do not need a calculator. All right, no calculator needed. No calculator permitted. Pencil, paper, that's what it'll be. Any other questions about the exam? It will be through Chapter 6, so all the way through. Okay. And I will probably, any other questions about this? I'll send you a full version. This is an abbreviated version. I'll send you a full version of this on Icon and by mail so that everybody knows what's going on. But I wanted to start getting people used to the idea that some of you will have to go somewhere else for your exam. And I'm trying to keep it as simple as possible so we're not going to ping-pong back and forth. Any other questions about the exam at the moment? I know exams are stressful. I'm trying to make this as least stressful as I can, but there's just a certain amount of inherent complexity to that thing. Any concluding questions about Chapter 4 or myoglobin or hemoglobin or protein structure that you'd like to ask before we move on to DNA? Any generic protein questions? Okay. Well, the next 2 chapters are going to be talking about proteins as well, but somewhat different kinds of proteins. And today we're going to be talking about the proteins that are involved in the cytoskeleton. And on Wednesday and Friday, we're going to be talking about that huge class of proteins called enzymes, looking at an example in particular. But the cytoskeleton is kind of an interesting group of proteins because it was one that really wasn't properly appreciated until roughly 20 years or so ago. And yet, it provides a very large fraction of the mass of proteins that cells make. It can be as much as 25% of the mass of a cell is in the cytoskeleton. So it's not a trivial fraction of the cell. The cytoskeleton or cell skeleton does a number of different kinds of things. When you see a shape of a cell, that's going to be provided for through the cytoskeleton. Almost all forms of movement of cells is mediated through the cytoskeleton, whether that movement is the division into two cell parts, or whether it's the swimming that we see with single-celled protozoa, or if it's the crawling that fibroblasts do to heal a wound, or the crawling that cancer cells do for metastasis. The cytoskeleton also has an important role to play in mechanical strength of tissues and of the organism. So there are a number of components of the cytoskeleton that if they are mutated, the whole organism sort of falls apart or can't stand up to things. Another aspect about the cytoskeleton that wasn't appreciated initially, perhaps, is that the cytoskeleton is one of the major markers for differentiation. That a good pathologist can look at a cell and say, oh, that's a liver cell, or oh, that's a muscle cell. When they're looking at it like that, what they're seeing is its cytoskeleton. All the enzymatic differentiation that makes it possible to do the work of a liver cell or the work of a muscle cell, that's there, but what they're seeing to make that initial identification that hallmarks it as this cell or that cell, that's cytoskeleton. And finally, if none of this is enough to make you intrigued by cytoskeletons, the cytoskeleton goes wrong, goes very, very viciously wrong in cancer. And it is disruptions in the cytoskeleton and its behavior that is one of the markers for how bad a cell has gotten as a cancer cell. And I'm just offering you here kind of a overview of the systems. There are three major systems in the cytoskeleton that are well documented. And often they're talked about as if they're completely separate things, which is misleading in most cases. In some cases, it's profoundly false. And I like this particular trio of pictures because it's one that lets you see the three filament systems in a single cell at once. And so this one is giving you an example of the actin filaments, and in this case, they've been bundled together into stress fibers that are very good at resisting tension on it. Here you have the microtubules of the cell, which are extremely long, slender structures that are good at bearing compression. And here you have vimentin, which is one of a member of a family of proteins that's good at giving elasticity as a kind of mechanical strength, a kind of passive mechanical strength. Okay. So let's talk a little bit about microfilaments first. This was the component of the cytoskeleton that was understood earliest, probably, and we continue to learn more about it almost every day. They're called microfilaments because they're thin, typically 7 nanometers across. And this is showing you here an electron micrograph of a thin filament, of a microfilament or an actin filament. Those are all synonyms. We find a lot of microfilaments underneath the cell membrane in many different cell types. And it may be as many as to almost look like a little felt there under the cell type. But we also find them in bundles or in cables, as if they are being knitted together to make little tiny cell muscles. And actually thinking of the actin filaments as an analog of muscles is not bad. Muscles is where actins were first isolated from. And the way in which microfilaments are made is kind of nifty. It has subunits, as so many things do. That subunit is a protein called actin. And this is a ribbon diagram of an actin molecule. And it has a binding place in it for ATP that can bind either ATP or ADP, adenosine diphosphate. And then these actin subunits bind together to make a helix of protein subunits. The individual subunits are globular, but because they pack together in a way, they can twist around and make a helix of them stacked up together. Sort of if you think about a bunch of Legos that's sort of close to square, but you can make them stack up and twist them around, and you can have a long, skinny stack, each piece of which is a little, mostly globular thing. When it makes that polymer, we call that an F-actin or filamentous actin. The subunit then is G-actin or globular actin or the globular monomer. And this is just showing you a space-filling diagram. So this would be one monomer, this is another monomer, and so on. So this is two strands of monomers that have twisted together to make the polymer. Now, the actin filament is not just helical, but it is polar. It has a handedness to it, or a head and back to it. And that's because the subunits inside the F-actin are all packed in, pointing the same way, that each individual subunit is pointing the same way. So in order to get that kind of a structure started, you have to have a couple of things together to make the rest of it on. And that initial process we call nucleation, getting a nucleus of 2 to 3 subunits to stay together long enough to make more F-actin. And so the nucleation process goes relatively slowly, more slowly than the elongation. Once you have that nucleus, you can elongate from that nucleus much more quickly. You've got something to add them to, and it's just going to throw them on there, and it's going to grow. And we call the actin filament a polar filament for this reason, that if you look at a bit of actin, so here you've got a stretch that's got 10 monomers in it. All the monomers are oriented the same way. Their ATP binding cleft is all pointed the same way. So this end is structurally different. It's structurally different than this end. And the structural difference shows up in a kinetic difference, that they behave differently. That wasn't a necessary conclusion, but it is a fact, that they are structurally different and they behave differently. So onto one end of it, new monomers can add, but somewhat slowly. And onto the other end, monomers can add, but can add quite quickly. So you have a difference in the rate at which the two ends polymerize. And you can see here that you start off with this original nucleus here, and the new part is much longer on the plus end or the fast end than it is on the minus end or the slow end. And if you kept on letting it polymerize till you used up most of the subunits, you'd end up at a point where they were adding to this end at the rate that they were falling off of that end, that they're not actually making new net filament, but they're bouncing off. And that shows up here, that if you let the polymerization reaction go to completion in a test tube, you'll have some point at which you have addition of subunits at the plus end and net loss at the minus end. You may occasionally get one come on, but you're getting more going off than coming on. And so overall, the overall effect of that is that the filament length will stay the same, but one end will have relatively newly added subunits, and the other end will be exposing the older subunits. And this process is sometimes called treadmilling, when the growth at one end is equal to the loss at the other. Treadmilling unquestionably happens in the test tube, but when you look inside of a cell, you don't see free ends of actin filaments very often. There are a lot of proteins that are involved in binding to those ends to control processes of polymerization and depolymerization and so on. And so we can see that there are proteins that can cap a fast end or can cap the slow end. There are proteins that can bind to the side of it. There are proteins that can branch it. There are proteins that can split it into pieces. There are proteins that can bundle it. So the actin filament by itself is just the beginning of a whole little zoo of actin-associated proteins that together give very rich and very complicated structures and behaviors. We do know that, for instance, for a cell to crawl forward, it has to be able to have new actin polymerization. So there has to be a way to get at that end to add in more actin monomers. Any questions so far? Okay. So is it all that important that cells can crawl? Well, yes. We see cells using here. So this is like a felt of actin filaments here. This cell is moving in this direction. So it has to have actin polymerization going on there to move forward. And that movement is important, for instance, during embryogenesis, when a small mass of cells has to elaborate and create organs and tissues and eyes and toes and all the rest of it. Cells continue to crawl throughout development at different aspects of development. Even in an adult, if you accidentally cut your finger with a knife and you have a cut, for that wound to heal, there are fibroblasts that will crawl to the correct place in order to close that wound and to restore the integrity of the skin. And then the scariest aspect of crawling is metastasis. that cancers can be divided into ones that stay as one mass and the ones that have shed new cancer cells to the rest of the body. That's metastasis. And in general, metastatic cells need to do a certain amount of crawling to get out and to get into the next tissue. So rapid crawling is often associated with metastatic cells. And this is just giving you a picture of how the structure of the cell reflects this function, in this case, of crawling forward, that this is an end where there's rapid change going on, and it's going out to explore the future. And this is the back end, the tail of the cell, and it will let go at some point and pull up and move forward. Actin alone cannot do too many of the things we've talked about so far. It cooperates with other proteins. And one of the important proteins with which it cooperates is a motor protein called myosin. And it's a motor protein because it's going to be able to use ATP, to bind ATP and to hydrolyze it, and to use that process to do mechanical work. So it's turning chemical work in ATP into mechanical work of actual movement. And if we look at the structure of myosin, we can see how the structure of the protein reflects that function. There's a long tail to it that is a coiled coil. We'll talk a little more about that in a little bit, but it's alpha helices coiled around each other. And then it has these two heads that are capable of binding ATP and light chains sort of in around there that are going to modify that behavior. And these myosin molecules can walk along the thin actin filaments by cycles of ATP hydrolysis. So the head structure is involved with the ATP hydrolysis and the motions. The tail structure is involved with making what's called a thick filament. Remember I called them thin filaments before. Here we have the protein that goes into thick filaments. And so all those little beads you see along this thick filament are myosin heads, each one of which can interact with an actin filament to generate motion. Here you can see an individual molecule. This is one individual protein molecule visualized in the electron microscope. So this is not just deduction. To some extent, we can actually see these things. Here we're seeing the myosin head in a bit more detail. It has this flexible neck region. It has an ATP binding pocket. Remember we talked about actin. It uses its actin to control polymerization. Myosin uses ATP to control mechanical movement. It also has an actin binding site. And the particular organization of the myosin and the actin is going to be the basis for making appropriate structured motions. And many of you may have seen the organization of actin and myosin in what are called striated muscles. So striated muscles are the skeletal muscles you stand with, your heart. And in them, what you see are thin filaments, actin filaments lined up parallel to each other, and then thick filaments of myosin interdigitated between it with their heads sticking out where they can interact with the actin filaments. And under the appropriate signal, they go through cycles of that walking process. And as the myosin walks along the actin, it slides further and further along it, but the space between the actin filaments gets shorter. So that's the contraction of muscles. No one protein is getting shorter. It's that the interdigitated structures are sliding past each other, and the muscle as a whole is getting shorter. And then under, again, the appropriate signal, they let go, they slide back out, and that's relaxation. Any questions about that? Okay. So myosin was one of the first places where what was called mechanochemistry was explored. And it looks something like this. So you have the myosin tail, the coiled coil. You have a neck and a head. It's touching an actin. If an ATP molecule is bound to that myosin head, it will now release its binding to actin. And as it hydrolyzes the ATP to ADP and PI, it swings. And so the ATP hydrolysis is coupled to a change of shape. In that conformation, it can rebind to an actin filament. And when it exchanges that bound ADP and PI for, releases it rather, it flinches back, it flexes back, and now you've restored the initial position that you had up there. So this is a cycle of bending, and releasing. This kind of cyclical motion is involved in cell division, making sure you've got the two parts of the cell correctly there, the kinds of cell movement I've talked about, muscle contraction, but also even intracellular transport. There are some parts of the cell that are moved around through myosin-related motility. Okay. These are some pictures. I don't know that we have time to go out onto the web and take a look at them, but they're some nifty examples of where you can actually see these things in real life now, that you can fluorescently label proteins in living cells through fluorescent proteins, and you can look at either tubulin in a living cell or actin in a living cell. Here's an epithelial monolayer. So these are all movies that allow you to see some of this cytoskeleton doing its actual job in real time. Slightly speeded up. So that's actin and microfilaments. That's one of the three major filament systems. It's especially involved with generating tension and with contraction. The second major system are the microtubules. And microtubules are, oddly enough, tubular structures. You know, if you think about the cardboard tube inside of paper towels, you're starting in the right direction. And if you think about that tube inside of the paper towels, it can resist compression. You can push against it, and even though it's just a little bit of cardboard, it resists that. And so microtubules are going to give structures that, in general, confer rigidity one way or another, that can bear compression. Microtubules, like microfilaments, are made up of globular proteins. So even though they're both long, skinny structures, they're made up of globular proteins. But by contrast, tubulin is a dimer. It's found as an alpha-beta dimer. And the nucleotide that is bound is different than actin. The tubulin proteins bind GTP and GDP instead of ATP and ADP. So they both use nucleotides, but they use different nucleotides. The alpha subunit simply holds on to its GTP. It just stays bound. The beta tubulin can hydrolyze the GTP and then exchange it after the fact to restore it. And because the tubulin dimer is oriented with a particular orientation, and because all the dimers in the tubule have that orientation, microtubules, like microfilaments, are polar structures. And so this is, you'll see the parallels here to what we were seeing with the actin thin filaments. There has to be some kind of a nucleus for things to get started. Whether that starts off of protofilaments or whether, as it is in the cell, there's probably a ring of a special kind of tubulin will vary, but there needs to be a nucleus of some kind. And in this microtubule structure, all the alpha and beta tubulin monomers or alpha-beta dimer, or each of the monomeric proteins, the alpha-beta dimers are all pointed the same way. And you can pick that up from the coloring scheme that they've used here. This particular sequence that they're showing you here of having a string of alpha-beta dimers or tubulin dimers and then extending it into a sheet that closes, that's one possible pathway in the test tube. In the cell, there are very specialized nucleation sites that control the growth of microtubules, and those are more likely to be actual rings to begin with, and then you can build more on top of that, like those guys who build up, beer can columns and stuff. Maybe that's a good image to have in mind, that you want all the pull tabs on the same end, or the thing won't be very stable. Don't ask me how I know. In all things, if you build something, at some point you have to get rid of it. General rule of biochemistry. And so we talked a little bit about the fact that actin can depolymerize when it's not needed. Same thing in the case of microtubules. One of the ways in which you can study that is in the test tube. So if you have a suspension of microtubules and you dilute it, you're likely to see that the ends of the microtubules sort of fray away from each other now that there's fewer subunits around to grow on. One of the things that's telling us is that there are side-to-side associations that are strong. We typically see in cells that the minus end the end that doesn't grow as fast as the other end, is usually anchored. And the free end is typically the faster or the plus end, the one that polymerizes more easily. It may also have proteins associated with it, like the microfilaments, so that whether or not it's an actively polymerizing end or not is also, to some extent, under cellular control. Like the actin, there are also associated proteins that can bundle the microtubules. There are proteins that can cross-link to make a looser kind of a bundle of things. And interestingly, antimicrotubial drugs are very good anti-cancer drugs. And a couple that you may have heard of are vinblastine and Taxol. These are both drugs that interact with the subunits of microtubules. And they have the effect, in the case of Taxol, of stabilizing the microtubule so it can't depolymerize. And this turns out to interfere with cell division because there's a cycle of making and getting rid of your microtubules there. Vinblastine tends to simply pull out of the population the free tubulins, so it's deposited in paracrystals that aren't available for good use. Okay. This is just some pictures to show you some of the structures of colchicine and of Taxol. Taxol was initially a drug derived from cypress or from cedar and had to be isolated from a natural product. But we've since then learned to synthesize it from scratch. It gives you kind of a sense for how complex some of these molecules are that are going to bind to them. Okay. So it talked about actin microfilaments being polar and being made out of globular subunits, and they had motor proteins associated with it. Microtubules are built out of globular proteins. They're polar. They have their own distinctive motor proteins associated with them. And so because there are motors that can associate with microtubules, things can move along microtubules to get to places. In the case of actin filaments, we were using the motor proteins for contraction. In the case of microtubules, it's more a matter of delivering things to places. And so some of the sort of dramatic things that get taken along microtubules are chromosomes. The whole act of mitosis depends on motor proteins interacting with microtubules. And like myosin, microtubule motors have a direction that they will move along the filament because it's not a symmetric structure. This is showing you a picture of kinesin, and kinesin looks somewhat similar to myosin. Again, a head that can interact with the microtubule. In the case of kinesin, the tails are on the other end, and you again have this extended tail of the coiled coil region. In the case of kinesin, these tails, the light chains on the tails, can attach cargo to that tail that can then walk along the railroad track of the microtubule to get somewhere. This gives you a little bit of a sense of the degree of organization of microtubules at the time of mitosis, that they're going to be doing a lot of work to get those chromosomes to the right place. So here's your ribbon diagram of kinesin. Again, you can see a compact domain in the head region. You can see a coiled coil in the tail and a kind of a floppy hinge region in between. And kinesin is kind of cute because it has a slightly different way of pulling off the trick than the myosin does. The heads do indeed interact with the microtubule, but they never completely let go. They kind of, like someone who's walking on slippery ice who doesn't ever quite want to let go of the thing that's holding them up. And here's that demonstrated in a schematic format here. So here's your cargo hanging off the tail of kinesin, your two different heads of the kinesin molecule. This head is bound already to the microtubule. When the ATP is bound at that head, it triggers a change in conformation to flip the other head forward. When the ADP is released from that second head, it can now bind to the surface of the microtubule, and now you have both of the heads in contact with the microtubule. The trailing head that was on it to begin with will hydrolyze its ATP. So ATP hydrolysis is the chemical energy that's going to drive all this mechanical work. And when it loses its inorganic phosphate, it now lifts up off the surface. And so you can see at every moment, the kinesin molecule has at least one point of contact on the microtubule. And so What that means is that movement along microtubules is processive. Processive is a word for a biochemical process where you do it and you do it again, you do it again, you do it again, you do it again along the same substrate. And we're going to see a number of processive things as we go through biochemistry. This is our first processive enzyme. But because kinesin is very processive, it can go through 500 or 1,000 cycles before it falls off. It means that microtubules make a wonderful railroad track for delivering vesicles to the cell periphery or delivering neurotransmitters to the end of a neuron and so on. Questions? So we've talked about two families so far, the microfilaments or actin filaments, the microtubules made out of tubulin, and then the third big group are the intermediate filaments. So the intermediate filaments are intermediate in thickness between the thick filaments of myosin and the thin filaments of actin. And the intermediate filaments, unlike microfilaments and microtubules, are very diverse, very diverse. Actin and tubulin are among the most conserved proteins out there. The intermediate filament proteins have 5 major families, and within those major families, there's a lot of variation. This particular picture is of a keratin intermediate filaments. This is in epithelia, so the skin that cover our bodies derive a lot of their strength from the intermediate filaments of keratin. And in general, what we're going to see is that intermediate filaments have a structural role, and they resist mechanical stress. They're good at being pulled on and not falling apart. Intermediate filaments are not polar. We talked about actin and microfilaments and microtubules having a plus and a minus end. Intermediate filaments are symmetrical from one end to the other, and we're going to see structurally why that is. Intermediate filaments also do not bind nucleotides. They do not need ATP or GTP in order to polymerize. They polymerize just inherently out of their own gumption. They form coiled coils where the one alpha helix is coiled around another. And that's the basis for their polymerization process. We also see a lot of places in the cell where intermediate filaments are cross-linked to either microfilaments or to microtubules. There's a lot of things that the cell does where 2 or all 3 filament systems actually cooperate to get something to happen. And I've used that word coiled coil a couple of times now. Let's take a little bit more of a look at that in detail. We talked about the coiled coils in the myosin tail and in kinesin. Intermediate filaments are almost all coiled coil. They're 90% or more coiled coil. And it's a structural motif that we're going to see in many different contexts. And so what a coiled coil is it's 2 alpha helices that have twisted around each other. So it's a helix of helices, if you like. And if you were to look at that in a little bit of detail, suppose you were to look down the top of an alpha helix. As you look down the top, you can see the side chains sticking out. And if you remember, there's roughly 3.5 residues per repeat. So if you went up 2 full repeats, you'd be back to the beginning of a turn. And that's what this diagram is highlighting. So we're looking down through an alpha helix here. We have residues 1, 2, 3, 4, 5, 6, 7, and it's going to repeat. And in a coiled coil, the residues that are on the inside of that coiled coil are both typically hydrophobic. And so if you look at the primary structure or the primary sequence of a coiled coil protein, you'll typically find hydrophobic residues at the 1st and 4 position in a repeating pattern of roughly 7-handed things. Now, the individual alpha helices of a coiled coil are themselves right-handed helices. That's what alpha helices are. But when you twist 2 of them together, the resulting helix is a left-handed helix. That is, the coiled coil is a left-handed helix. And these 3 representations just give you slightly different ways of seeing that characteristic of it. They've coded one of them with green and one with blue. You can see that they're each of them alpha helices twisting around each other. You can see here side chains sticking off. And here I think it's more clear that it's a left-handed helix. You can see it twisting up to the left. With intermediate filaments, two intermediate filament monomers make a coiled coil or a dimer. If you take two dimers and stagger them, you can make a tetramer. So here's a dimer of coiled coils. And in the case of intermediate filaments, they are parallel coiled coils. They have their N-terminus and their C-terminus together. But when you pack them together into tetramers, they now are packed anti-parallel. So there's no head or tail to the filament overall. Because the tetramers are packed anti-parallel, you can't tell which end is which. They're equivalent. They have the same physical characteristics. And then these tetramers can bundle into octamers and so on until you end up with the intermediate filament that is a bundle of a number of these. Intermediate filaments are very insoluble. They are, some of them, so insoluble that you almost have to degrade some of the proteins to get them into solution. Others have to be highly phosphorylated before they're released and so on. But they're intended to be insoluble. They're intended to be stable structures. You don't want your skin going away when you go in the bathtub. There is no polarity to the filament overall. There is no nucleotide binding and no nucleotide hydrolysis. This coiled coil structure is entirely intrinsic to the primary sequence of the intermediate filament protein. You might ask yourself, how do you assemble something that's so insoluble? And some work from our laboratories showed that in some cases, vimentin actually is assembled where it's translated. Since you can't really solubilize it very easily, maybe you put it together where you want it to begin with. And as a consequence, its messenger RNA is localized to the sites where that assembly is happening. So it's sort of a side note. Okay. So any questions about that so far? Those are the three major cytoskeletal systems. Each of them has associated proteins with them to make them more complex, and they interact with each other. Any questions on that before we talk about collagen a little bit? Okay. Collagen is a structural protein, but it's an extracellular structural protein. The three families that we've talked about so far in the cytoskeleton are all inside the cell. Collagen is outside the cell. And collagen is another good example of how structure reflects function. It has a very strange composition. You may remember back when we were introducing you to the amino acids that proline is weird, that proline has this closed ring structure instead of a free-flopping side chain. And because proline closes around on itself, where proline is inserted in a primary sequence, it's not quite as free to rotate and move around as many other residues would be. It constrains it to some extent. Collagen has a lot of proline and a lot of something called hydroxyproline, where this carbon on the proline is hydroxylated after the protein is synthesized. That hydroxylation requires vitamin C. That's why if you don't have vitamin C, you get scurvy. You can't hydroxylate your proline to hydroxyproline. Okay. So if you take a look at collagen and how it packs, it has this odd kind of a very extended helix. It's a left-handed helix of the backbone itself, of the polypeptide. And in the case of collagen, you need 3 polypeptides to make a triple coil. A little strange. And so here you can see the 3 that have been sort of woven together. It's a little hard to see in this one that it's in fact now a right-handed helix of the triple coil. So it's the opposite of the coiled coil that we saw in the intermediate filaments or the myosin tail or kinesin. In this case, the fundamental or the core is a very extended left-handed helix twisted together to make a triple right-handed helix. And then if you get lots and lots of copies of this triple helix, they will then associate side to side with each other and make collagen fibrils. And so this is electron micrograph of collagen fibrils where with staining you can see the periodicity of it, the fact that it's a very regular structure. And these kinds of collagen fibrils are the things that make tendons so strong, They're an important part of the extracellular matrix to bones and are part of what make bones resilient and not just brittle, and so on. Now, the trick to collagen and why collagen forms a triple helix and not something else is in its sequence. So the primary sequence in general is, I mean, with some exceptions, but there's a lot of this in it. Glycine, proline, hydroxyproline. And when you look at it in its native state, you see it makes this very extended left-handed helix with the glycines all tucked into the middle. So this is another example where glycine, as the smallest of the amino acids, is the only one that can fit in there. So if you look at this structurally, you can see how trying to pack in any kind of a side chain in there, there wouldn't be room to do it. And then the proline and the hydroxyprolines on the two outer limbs are probably contributing to the fact that collagen as a protein doesn't rearrange very easily or very much. The chemistry of collagen goes beyond that still, so that in addition to the need to hydroxylate the proline to make hydroxyproline, there are some other residues in there. It's not just those three. And amongst those, one of the residues that occurs up with some frequency is lysines. And these lysines can become covalently cross-linked to each other. So you can end up with a covalent cross-link between two collagen molecules. So you can imagine why, structurally speaking, collagen is such a great thing for building tendons and so forth out of. It has this great spring structure to the protein itself, and then in the bundles you're adding additional layers of covalent cross-linking to go on with that. And does that stage also require vitamin C? Do you remember, Eli? You're not sure. Okay. That's fine. But the fact that vitamin C is used for the hydroxyproline formation is why you start to get bleeding gums and things without enough vitamin C, because literally your collagen is starting to break down. Any other questions then before we say hello to enzymes on Wednesday? Okay. AV, do you want to turn off the recording?